Welcome to our next video. In this one we're going to talk about proteins, and this is section 2.4. Again, this is for the 2016 starting IB Bio exams. And in proteins, one of the main things that we need to talk about, obviously, is amino acids, because amino acids are the building blocks of protein. They are what make up proteins. If you think back to carbohydrates, the monosaccharide is a single sugar that helps to make up a larger polysaccharide. Well, amino acids are the same deal, a small piece that put together makes up a protein. There's about approximately 500 known amino acids, but only 20 of those amino acids are used in building proteins. And amino acids have a very similar basic structure or shape, and this is it. We'll, on the next slide, take a closer look at this. But each has each amino acid has a similar basic structure, and then one different thing that's attached to it that changes, and that's the R group. So let's take a closer look at that. Here's a nice image that shows us the basic structure of an amino acid. Each amino acid has an amino group, which is a nitrogen with 3 hydrogens. That's connected to a carbon that has a hydrogen and an R group. We'll talk about that in a second. And then lastly, a carboxyl group. This actually got cut off in this video here, but this carbon is double bonded to an oxygen, and then this oxygen has an H to attach to it. It should be an OH. The ribosomes in a cell are what connect these amino acids together to form a protein. So a protein is a bunch of these amino acids put together in a sequence to make a protein. And that sequence is stored in the genes or the DNA. Genes are a section of DNA, and that section of DNA, that gene, codes for or provides the instructions to make specific proteins. What makes each amino acid different is what is attached here as the R group. And it could be as simple as another hydrogen being attached, or a CH3, a carbon and 3 hydrogens. What is attached here can change, and how whatever is attached there makes that amino acid different. Life shares these 20 amino acids, and they're ideal for making a wide range of proteins. Sometimes these amino acids can be slightly modified. For example, in collagen, the amino acid proline, that's a specific amino acid, the proline is the specific amino acid, it's converted to hydroxyproline. And this makes the collagen more stable. Really, only a small portion of genes are used to make proteins, and this small chunk of DNA, this small section, the gene, that is actually used is called the open reading frame. And so as you can think about the different combinations that could be put together. If there's 20 different amino acids that are used for making living things, the order or the sequence of how those are put together, the possibilities are not endless, but nearly endless. There's a lot of different possibilities of how these amino acids could be put together to make different types of proteins. So the next question is, well, how do these amino acids get put together? And that is something that we've already seen. That's through condensation and hydrolysis. We've already talked about this with both carbohydrates and lipids. And a quick recap of this: condensation reaction, 2 molecules combine to form a single molecule, and it results in a single molecule and a small molecule. We can call this dehydration reaction if that small molecule is water. We're going to look at this with amino acids here in a second. Hydrolysis reaction, the breaking of a single molecule into 2 smaller parts by using water. And so what this looks like, here are two molecules here. These are carbohydrates, and this is sucrose that's being broken down by hydrolysis. Water is being added, and we get glucose and fructose. In this case, condensation, water is being removed from 2 amino acids to make a bonded or connected amino acid, 2 amino acids together, and this is called a peptide linkage. Let's take a closer look at this process for amino acids. So we have 2 amino acids. Here's our 2 amino acids right here, 1, 2. And what is removed is the OH from one carboxylic portion of the amino acid. So this OH is removed, and one of the hydrogens from the amino group is also removed. And so we get water, H2O, we get water. And then how these are connected is between the carbon that has the double bonded oxygen and the nitrogen that only has 1 hydrogen. Both the nitrogen and the carbon are happy or stable because they have the right number of bonds. Carbon has 4, nitrogen has 3, and the linkage, or the way that it is connected, It's called a peptide bond, and they're connected together. That creates both this protein, this dipeptide, and water. If more and more amino acids are added, we could have another amino acid here on this nitrogen or this carbon. If more amino acids are added, it makes a polypeptide, which is a chain of amino acids, and a polypeptide is a protein. If you ever watch The Office, Dwight Schrute is very adamant about referring to protein as polypeptide chains, and he is correct. It is a number of peptide bonds put together that make up a protein. The structure of proteins is not a 2D structure, but really it's a 3D structure. As these individual amino acids start to get put together, they can be folded and combined into different shapes. And two different shapes that we see is a fibrous protein, which has an elongated repeating structure, or a globular protein. And this has a helical or sheet-like shape, and we'll look at these in a second. The folding of proteins, of these amino acids put together, helps to stabilize the protein, and this is accomplished oftentimes by hydrogen bonds. We talked about hydrogen bonds in our water video. Well, this is one of the first other examples of hydrogen bonds. They help to stabilize the folding of the protein. The 3D structure is really determined by the amino acid sequence, and so we start to see that a little bit here. These are all different amino acids, and so here we can see a hydrogen bond, and that's the structure of it is helped actually determined by the sequence. So these two amino acids are hydrogen bonding between one another. This is a primary structure. This first is just a primary structure, and what's happening in a primary structure is simply just the linkage of these amino acids together. So it's a polypeptide chain. That is a link of a bunch of amino acids together, and that makes up the primary structure. A secondary structure is when this link of amino acids put together starts to actually take some shape. And it can take the two different forms of shape. It can take an alpha helice, and this is very similar to like a double helix, except it's just a single upward spiral chain or link, you could think of these amino acids together. Again, hydrogen bonds helping to stabilize. Or it can form a beta pleated sheet. And this is like if you were to take a piece of paper and fold it, if you were to fold it multiple times and then pull it apart, it would have up and down kind of ridges. That's kind of like what this beta pleated sheet looks like. And you can see in both cases there are some hydrogen bonds here shown by these dotted lines that are helping to stabilize this. That is secondary structure. Tertiary structure is then the actual complete folding of the protein. And this doesn't always have to happen. Sometimes the proteins are actually not folded into a specialized shape, but oftentimes it does. The last and final is the tertiary shape, quaternary shape or structure, and this is when multiple of these structures are being put together. Multiple different proteins are being put together, and hemoglobin is a good example of this. Hemoglobin being what's used in blood to actually help transport and carry that oxygen. Here's a nice image that shows all of these put together: primary structure, secondary structure, tertiary, which is our third, and quaternary structure. We can see just our link of amino acids here. In our secondary structure, we see our alpha helice, our beta pleated sheet. Our tertiary structure, the folding of that protein, and then quaternary structure is multiple of these proteins put together. In looking at the different structures and different types of proteins, we've got fibrous, as we already mentioned, and globular proteins. The fibrous proteins are insoluble in water. They have a longer, elongated shape. They're physically tough. Collagen is a good example, found in both skin and keratin, and keratin in hair is another example. These are more tough proteins. Globular proteins, on the other hand, are more compact in shape. They're more folded together, more condensed. Fibrous is longer and stretched out. Globular proteins are soluble in water. They have a rounded shape. An example would be hemoglobin and enzymes. Those would both be examples of globular proteins. In some situations, proteins can be denatured or broken down. And denature is a protein being denatured is a permanent change in the protein due to breaking of bonds and interactions between the R groups, the portion of the protein that can be variable. And some causes of a protein being denatured is heat or pH. What happens when heat is applied is it causes vibrations within the molecule that can break the intermolecular bonds or interactions that help to hold it together. Proteins have different heat tolerance, so different heat levels will cause different changes, or it will take more heat or sometimes less heat depending on the protein. The cooking of an egg is a good example of this. You can fry an egg, and as the yolk starts to turn from kind of a lighter orange color, yellow color, and as the white portion starts to, the clear portion actually starts to turn white, that is the proteins being broken down, being denatured because of the heat. pH, or potential hydrogen, is also another way that proteins can be denatured. And extreme changes in pH can cause the denaturization of proteins, and this also causes changes on R groups and the breaking of bonds that help to hold that together. The stomach, in our human stomach, we have a pH of about 1.5, and that is the optimum pH for the protein digesting enzyme pepsin. Pepsin helps to break down the food that you ingest, and the optimum pH for that enzyme, that protein to work, is about 1.5. And so inside of your stomach is very acidic to allow for those enzymes, pepsin specifically, to help break down food. The last topic for this video is proteomes, and these are all of the proteins produced by a cell, tissue, or an organism. What is interesting or unique, I guess, about this topic is that the genome of an organism is all the genes of a cell, a tissue, or for an organism. So if we're looking at this at an organism level, the genome would be all of the genes for that organism. Proteome is going to be all of the proteins produced. The genome is fixed, meaning it can't be changed. I suppose mutations could cause it to change, maybe exposure to radiation. But the proteome is variable. The genome is fixed, the proteome is variable. The proteome of each individual is unique due to differences of activity and small differences, mutations of the amino acid sequence. Each person has a unique proteome, including identical twins with the same age. So identical twins are going to have slightly different proteomes. In this video, we did not look at specific protein functions, nor do we look at some specific examples of proteins. You can find those on the internet somewhere, or in class we were going to go over those together, so you'll have a chance to look at some specific proteins and some examples of functions of proteins in class together.